Okay, this is Mr. Russell, your AP Biology teacher, and today we're going to be talking about basic cell structures. We know that the cell is considered to be the basic unit of life, and we know that cells are examples of emergent properties. You think about emergent properties, think about a puzzle. If you just have one piece of a puzzle, you may not know what the outcome is going to look like. But when you put the pieces of the puzzle together, they will converge and fit together, and then a picture will emerge out of that. So then once you've got the puzzle completed, you will have emergent properties. You have an image that will show up. Now if we think about cells and organelles, if you only have organelles, then nothing can happen. But if you have all the organelles together and inside a membrane, then life can emerge from those cells. All cells are considered to be open systems in their natural settings because there are materials coming into the cell from the surrounding environment, as well as materials leaving the cell and going into the surrounding environment. So the cell is an open interaction with the environment. Well, cytology is the study of cells, and a person who studies cells is referred to as a cytologist. We're going to look at two types of cells. That's going to be prokaryotic cells and eukaryotic cells. Prokaryotic cells, pro means before, kary means nucleus, ot means organisms. So prokaryotic cells would be a before nucleus organism. So think about bacteria as a good example of prokaryotic cells. It's thought that it would have evolved before a nucleus would have. And it's believed that these prokaryotic cells would have come into existence about 3.5 million years ago. Evidence of these, the oldest prokaryotic fossils are found on stromatolites or bacterial mounds in Shark Bay, Australia. And here you can see a picture of that. If we think about eukaryotic cells, animal cells, or plant cells, these cells would have truly come after a nucleus had been formed. So all of these organisms, plant cells and animal cells, they both have a true nucleus. Now, Lynn Margulis, I believe is how you pronounce her name, she proposed what is known as the endosymbiotic hypothesis back in the 60s. It basically says that prokaryotes came to live together in a symbiotic relationship. A tick on a dog is an example of parasitism or a symbiotic relationship. Mutualism, where both organisms benefit, neither are harmed, would be something that might have happened here with these prokaryotes. So what she's saying here is that the smaller prokaryotes, they lived inside of the larger prokaryotes. They did this to gain survival and advantage over the other prokaryotes, and eventually it's thought that they evolved or changed into eukaryotic cells over many generations, which may have spanned thousands of years. Why did they do this? Well, it's thought that these smaller organisms, or smaller prokaryotes, did so to gain protection from the surrounding. So larger organisms allowed the smaller ones to live inside of them so that they could gain energy, and maybe it would help them move faster or have a faster motility. Well, over time, these DNA segments were swapped to create a more permanent existence, because we know that bacteria, they have DNA. This swapping is referred to as genetic annealing, with two N's there. Supporting pieces of evidence will be, we'll see this later when we look at the mitochondria, the chloroplast, and the flagella as well. Here we're looking at a picture of modern day prokaryotic cells, animal cells, plant cells, and then how we see them today under microscopes. And here we're looking at references for size between viruses, bacteria, and animal cells. Viruses obviously very, very small, but then bacteria are a little bit larger. But look at your comparison between your prokaryotic and your eukaryotic cells. Your animal cell, eukaryotic cell, is very, very large, has a large nucleus. It's going to be a good bit bigger. It's got a lot more organelles, a lot more information inside. Well, it needs room to be able to have that information inside. So you're going to see throughout this year that surface to volume ratio is of great importance for cells. So cells can be only so small. There has to be enough room or volume to hold things and to perform work inside a cell using the cell membrane. And in the opposite of that is that cells can only be so large. Larger means more traffic going into both directions, into and out of the cell across the cell membrane. So a cell must be large enough to obtain DNA and ribosomes and be able to make proteins, and some cytoplasm to act as a working space. They can only be so big because we have to be able to have enough room for food into and waste out of the cell efficiently. Got to be able to move it without things getting too clogged up or too backed up. If it is too large, the cells become ineffective or inefficient at moving these things, so it divides and goes back to a smaller state. So think of cells as cubes. We can find the surface area of a cuboidal cell by using the formula length times width times 6, or 6 times side square. Easiest thing for me to remember is surface area is length times width times number of sides, which is going to be 6 sides in a cube. We can also find the interior volume by the formula length times width times height. Or you could refer to that as length times width times height, or sides cubed. This is an easy measurement. You can do this and make sure you put down your proper units when we're doing these calculations in class: centimeter cube, millimeter cube, or meter cube when we're talking about the volume. We're talking about surface area, we're talking about centimeter square, millimeter square, or meter cubed. Now, the comparison is made as such. Surface area, you're going to put your colon and put volume. Because we're asking for surface area to volume ratio. If you look in your boxes there, actually I don't have this in there, and I'll show you this one in class as I need to. So surface area and volume is going to be the lowest common denominator. And for all cells, they desire a much higher surface area than volume so as to be most efficient in transporting by diffusion across the membrane. Here's one way I think about surface area and volume. You've got a bottle of water. On the inside of your water, or on the inside of your bottle, is your water, and that is your volume. Now, maybe you've drank a little bit of your water. Maybe you've drank half the bottle. If so, you have more working space in there. You could add more water. But what happens if your bottle is half full and then you try to pour another bottle of the same size totally inside of your bottle you're already working with? It would overflow and be a mess everywhere. The reason is because you don't have, you don't have enough surface area. Now, your surface area is going to be your outside container. So if you think about a cell membrane and a cell, the outside of the bottle would be comparative to a cell membrane, whereas the inside, everything that's living in there, is going to be taking up volume. If you move things out, maybe you take a sip of your water, you're moving water out of the bottle, so you're reducing your volume and increasing your working space. If you put more water back in, you're reducing your working space. Alright, now, so we think about prokaryotic cells, or bacteria. All prokaryotic cells or all prokaryotes, they are unicellular. The cells possess inside DNA in the nucleoid, not in the nucleus. They also have ribosomes, and they have a cytoplasm. So there are 3 basic shapes of prokaryotes that exist. One is coxy, meaning round. And here you can see that, how these look like little balls that are kind of linked together. Then you've got bacilli, or bacilli, which means rod-shaped. And then you've got your helical bacteria, which are going to be spiral shaped. So go back and take a look at those three again. You've got your round ones, your cocci, your bacilli, which are going to be your rod, and then your helical, which are going to be your spiral. Now, when we look at prokaryotic cells, they're going to have a cell wall. This is not the same type of cell wall that plants would have. This structure is mainly primary for protection of the underlying cell membrane. It's kind of like a water balloon. If you're filling up water balloons to have a water balloon battle, typically you might put those in a bucket or something, or in a bag. Why? Because you want to protect them and keep them from bursting. Well, this is kind of how the cell wall is acting. It's a protection for the cell membrane. It also helps to prevent the prokaryotes from bursting, like the water balloon, in an aquatic environment. You get too much water in a water balloon, it's going to rupture. Well, the cell wall will kind of help to reduce that. The cell wall is hypertonic to the surrounding hypotonic solution. Water travels hypo or hypotonic to hypertonic. That's one of those things to remember. The cell wall is mainly composed of proteins and sugars. When we think about proteins and sugars, peptides refer to proteins, and glycan refers to sugars. So when we're talking about something that's made up of proteins and sugars, we're talking about peptidoglycans. So we're looking at these terms. When you're looking at an exam, whether it be the AP exam or any of the tests that we may take, try to look at the term itself. If you don't understand it, try to break it apart and be able to synthesize what's really in the word. Peptidoglycan. We're not using that every day. If we know peptides are proteins and glycans are sugars, we can figure out that this is a sugar of proteins, or protein sugar. Now, scientists perform what's called a gram staining for easy identification of most bacteria. And this is what you're looking at here. A gram positive will stain blue. They possess a very thick peptidoglycan layer. A gram negative, which is going to be a red stained cell wall, these are going to be thin peptidoglycan layer between the phospholipid layers. Now, the problem here is these gram negatives, these are the ones that are more dangerous to us, and they're usually resistant to antibiotics. These are things like your MRSA, which you'll learn about in maybe a different health science class, and we're going to talk about this a good bit later in the year as well. Now, some bacteria can produce a capsule that covers the cell wall. This capsule is a very sticky substance for adherence to surfaces. A lot of times it's just the capsule material is what actually makes people sick, and not necessarily the bacteria itself. Here we are, we're looking at our prokaryotic cells again. We're seeing there that it has a nucleoid. We see that it's got the ribosomes, plasma membrane, the cell wall for the protection, and we've got this capsule for this outer part. So if we go back and we look at our picture of our animal cells and our plant cells, our modern day eukaryotic cells, we're looking at three main parts. It's got a plasma cell membrane that's going to help to hold the cell together. Well, let's look back. So does a prokaryotic cell. It has a plasma membrane as well that's going to help to hold the cell together. Now, when we look at our animal cells and our plant cells, or eukaryotes, they have a nucleus. This controls the activities of the cell by holding the DNA. The DNA is just the instructions, whereas a prokaryotic cell has DNA in its nucleoid. Oid meaning like, so it's like a nucleus. Now, if we go back and we look at our eukaryotes again, our cytoplasm or cytosol, this creates room for work and space for holding organelles and ribosomes. Now, if we go back and we look at our prokaryotic cells, then this is the same or very similar between the two types, between our prokaryotes and our eukaryotes. The only difference here is that prokaryotic cells do not have organelles. All right, so this wraps up with our basic cell structure. We will continue on our next session.